anaerobes, we ingest molecules, we're going to break those down. And we're going to basically go through the process of degradation of glucose in glycolysis and the synthesis of glucose in gluconeogenesis. Glucose plays such a central role, we cannot dispute that in regards to the storage of glucose in the form of glycogen, starch, or sucrose, and we'll see how we produce glycogen a little later on, as well as starches. We're going to see, importantly, how we take glucose and oxidize it through the oxidation potential reactions to produce pyruvate, which is then going to feed later on into another cycle called the Krebs cycle. We can also use glucose in the form of pentose phosphate pathway to form ribose 5-phosphates to produce other carbohydrates. But a carbohydrate, specifically these 5 ribose sugars, that will be bases for our nucleotides in regards to DNA and RNA. So again, pentose phosphate pathway plays an important role, but also it plays another important role in regards to sinking electrons and mopping electrons up in regards to the capacity to control radical species. And we'll have a little bit of a look at that. We're not going to talk too much about the issue of synthesis of polymer structures. If you want to learn more about that, you can go and do a plant biochemistry course, which is run by Dr. Owens, but it's a graduate class. So again, they talk about glucose production and then producing things such as xylulose and cellulose. And remember, just a recap of our structure of glucose and our naming constituency: 1, 2, 3, 4, 5, 6, phosphate 6. So in this case, this is glucose 6-phosphate, which is going to be the key component in our metabolic profiles. And here we have the cyclic form versus the linear form. But we want to, again, emphasize the issue of the single carbohydrate moiety and regards to the contribution in regards to osmolarity. Now, we want to make sure that we have carbohydrates available, but we don't want to have those carbohydrates sinking the situation of osmolarity and trying to make sure there's a balance. So let's go forward and start our investigation on chapter 14, page 533 to 558. So glycolysis versus fermentation is basically a switching system in regards to electron transfer and sinking those electrons into a system. So normally we see glucose through glycolysis to go to pyruvate. Now, pyruvate has a choice depending on the cell's capacity and its issue of oxygen availability, whether it goes to the pathway A to produce lactic acid or lactate, or path B, if there is oxygen available, to go through the process of utilizing the pyruvate to degrade it completely to produce carbon dioxide and molecular oxygen and give us that ATP requirement from the breaking of those carbohydrates in the form of pyruvate. This only happens depending on the oxygen capacity of the cell or availability of oxygen in the system. We have a low oxygen system, we are going to basically promote lactic acid formation like a sprint. So we're going to do a lot of exercise, we're going to need a lot of ATP, but we're going to get ATP coming from this system too, but it's not going to be the full utilization of the individual breakdown molecules in glycolysis. In the fermentation process, if we have low oxygen levels, we're going to produce other byproducts. And with fermentation, if you remember with fermentation, we can produce alcohol, which is going to be a byproduct given that there's less abundance of electrons and oxygen availability, more so the oxygen availability. So in this case, when we're looking at movement of the action potential and muscle propagational movement, as we've seen with actin and myosin, we want to see ATP coming from both systems, from the glycolysis as well as through the metabolic process of the mitochondria, and providing that particular energy. But this might change if we depend on our oxygen availability. So anaerobic versus aerobic metabolism is a very interesting area because there's always going to be a payback. We cannot have one without the other. And if we're doing lactic acid formation, we're going to bring this back to pyruvate to bring it through pathway B. We cannot store large quantities of lactic acid or lactate in our circulatory system. It's detrimental, causes pH changes, and causes damage to potential muscle fibers and proteins. So there's always going to be a payback, and we're going to see these seesaws moving through the system in regards to allowing us to understand, importantly, what is going on with our particular system. So let's have a look at glycolysis in regards to the two phases. We have a preparatory stage and a payoff stage. So glycolysis to pyruvate is 10 individual chemical steps, and we're going to know those 10 individual steps and be able to draw them. We're going to have a preparative stage, and we're going to have to consume energy. But we're going to get in the payoff. Remember, we talked about net gain. We're going to see that payoff invest some of that particular currency in the preparative stage and allow us to have an overall net gain of the 10 reactions from glucose to pyruvate. But at each individual step, we're going to have mechanisms of regulation in regards to controlling whether we need to go to preparative or payoff stages, or we need to shut down the whole system in regards to not consuming our glucose, because we might have too much energy in the system and no longer need any more pyruvate. So let's start. Here's our glucose molecule, as we can see with our positions 6 to 1. We're going to use a hexokinase enzyme, and note this is in one direction. So we have our first point of regulation control, a commitment stage in regards to this particular enzyme doing a job with magnesium as well with ATP, and degrading that ATP to form ADP and phosphorylating this number 6 position to give us glucose 6-phosphate. And in this case, the delta G prime 0 is minus 16.7 kilojoules per mole. So it's in the positive direction. We're going to be able to move this quite happily forward in regards to the phosphate phosphorylation of the glucose with the energy coming from the ATP. So again, we have that degradation causing a phosphorylation on our glucose moiety. The next step is going to be then looking at the glucose moiety in its glucose 6-phosphate using phosphohexokinase isomerase. And this particular guy is going to take glucose 6-phosphate and reconfigure the glucose 6-phosphate into a 5-membered ring, and basically now giving us a position of carbon outside the ring in position number 1 and position number 2. So we see this position being rearranged so it's outside the ring, and this produces then fructose 6-phosphate. So here we have fructose 6-phosphate, and in this case, the 6th position is phosphorylated. We now have another utilization of ATP. So we've already consumed one priming reaction and now a second priming reaction, consuming that ATP to produce ADP to now produce fructose 1,6-bis, 2 phosphates at positions 1 and 6. So we now have this particular phosphate group being this particular hydroxyl group being now phosphorylated, giving us a symmetrical molecule, a near symmetrical molecule in regards to how we're now going to cleave this with an aldolase, which is a reversible reaction, to produce glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So I want you to show and very clearly note the mauve and the green constituents. So in this case, the glyceraldehyde 3-phosphate comes from carbons 4, 5, and 6, where the dihydroxyacetone phosphate comes from carbons 1, 2, and 3. They both contain phosphate because of this position in the 1 and the 6, and therefore we get the cleavage. But note again, the aldolase reaction is reversible. So we have one hexokinase and the phosphofructokinase, which are commitment steps because they are not reversible. And we're going to come to those non-reversible questions already. So we get 2 molecules of 1, 2, 3 carbons. So two molecules of 3 carbons as we break down our 6-carbon molecule constituent in the form of glucose. So again, I stress that carbons 1, 2, and 3 give you the dihydroxyacetone phosphate, and the carbons 4, 5, and 6 give you the glyceraldehyde 3-phosphate. So we have two molecules. Now, there's something going to be very interesting occurring between these two particular molecules, and it's through a particular enzyme. So what we see, let's go here, Oops, actually I was going to do a closer look in regards to glucose 6-phosphate. So think of our enzyme interaction. We're going to have a base pair reaction to get this particular position to become phosphorylated, and ultimately rechange the orientation of the molecule through the phosphokinase isomerase. In this case, the phosphofructokinase number 1 is the key component to give us this particular reaction. And in this case, we're going from glucose 6-phosphate to an intermediate to fructose 6-phosphate, we're going from a linear form. I show these in a linear form. And in this case, there is an energy requirement. It's not a exothermic exogonic reaction. It's an endogonic reaction. We need to actually supply the fructose kinase number 1 some energy, and we're going to do that because we need to satisfy this 1.7 kilojoules per mole in the positive. So we need to have some energy being consumed to allow us to actually get this reaction to go. Once we get to the fructose 6-phosphate, we can then add more ATP. So this is the commitment stage. This particular reaction step is going to take negative 14.2 kilojoules per mole. That energy is coming from the hydrolysis of ATP to make ADP, and that inorganic phosphate that is going to be incorporated into the glucose 6-phosphate to produce glucose 1,6-bisphosphate. So again, phosphate in the number 1 position that was now added from this ATP. The phosphate in the number 6 position was added from the first enzyme reaction in regards to reducing glucose to glucose 6-phosphate via the hexokinase. Now with the aldolase, which is a reversible reaction, we're going to have the dihydroxyacetone phosphate and the glyceraldehyde 3-phosphate. Again, indicating where those particular carbons are coming from the splitting of the individual molecule. But noting again, we have a high energy commitment state. We must find 23.8 kilojoules per mole to get this particular reaction. So you can see why we call this the preparative stage. We need to invest money, or energy, I should say, invest energy into this system to give us the capacity to allow this reaction and this reaction to occur. And again, we have some capacity to produce 2 3-carbon moieties. And the interesting thing is there is a triose phosphate isomerase, which has the capacity to change this molecule to make sure that we can either produce dihydroxyacetone phosphate as the major constituent or glyceraldehyde 3-phosphate as the main constituent. And we're going to need this guy. So we're going to transfer the dihydroxyacetone phosphate through the triose phosphate isomerase to produce more glyceraldehyde 3-phosphate. So the enzyme does the hard work of transferring the dihydroxy to acetone phosphate to make glyceraldehyde 3-phosphate, because this is the guy we are going to take on to the next reaction. We're not going to take this guy on. We want to transfer this guy to this, and we're going to then siphon this glyceraldehyde 3-phosphate to the next reaction. Okay, so I'm going to leave it there because we're at our time now, and we'll continue our reactions in regards to the degradation of our glucose moiety, which is now going to be illustrated in regards to what happens within the enzyme cleft to give us that dihydroxyacetone phosphate and that glyceraldehyde 3-phosphate. And ultimately, we're going to have to understand, importantly, where those carbons are coming from. As I said on Wednesday, I'll give you a small lecture on radioisotopes, because this is